Welcome! In this video we want to show you how to explore CovidGraph with SemSpect. You can see the interface of SemSpect here. The different node labels of CovidGraph are listed on the left. The blank screen in the middle can be used for exploration. To do an exploration, we're going to choose one of the node labels. We're going to choose Gene and then drag it onto the white canvas. You can see here that all the nodes with the label Gene are now grouped together. If we click on that group, we can open the node table. The node table can be used to filter. In this case, we're going to filter for genes with the SID PF4. We can now select our genes of interest and then restrict the group to our selection. Now that the selected group is a bit smaller, we can see the individual nodes inside the group. If we click on one of the nodes inside the group, we can tag it. Now we want to start our exploration. Therefore, we click onto the group and then select Genes and all relationships, thereby displaying all genes that are somehow connected to the genes that we have selected. In this group, you can see different synonyms of the 4 genes. We can now use this group to further explore the relationships of the genes. If we click on the group, we can select fragments, which are text fragments that mention the genes of interest. Fragments can be part of a body text or a patent description. And if we select patent descriptions, then we can further explore the patents that are connected to our genes. Each patent has, for example, a patent title, which we can also display. And again, if we hover over the nodes inside the group, we can see the different patent titles, and we can even tag the nodes. We can also display the owner of a patent by selecting Entity and then selecting the relationship owner. As you can see here, we can again tag individual nodes of the group. We can also explore the different papers that are connected to our selected genes. Therefore, we go back to the fragments, select Body Text, and then select Body Text Collection. Each body text collection is part of a paper, which we can also load here. And as you can see, there are different papers that are related to our selected genes. In addition, we can also explore the underlying biological connections. Therefore, we go back to the group of genes and then select Transcript. From there, we can go on to the encoded proteins. And these proteins are associated with different ontological terms. These ontological terms include, for example, platelet activation or heparin binding. We can now use these ontological terms to go further into CovidGraph. By selecting Masimoths, we can explore the systems biology part of CovidGraph. Lastly, we want to explore the different pathways where the genes are involved. We click again on the group of genes and then select Pathways. Our selected group of genes, for example, is part of the pathway hemostasis. If we hover over one of the nodes inside a group, you can see that all the connected nodes on the branch are highlighted in green. And we can tag all of these connected nodes on the branch.